Welcome to today's webinar. Stability Studies, the Architecture Behind Establishing a Beyond Use State is presented by Brian Kelly, Business Development Director for ARL Biopharma. We also have Dr. Thomas C. Kupik, President and CEO, and Vince Bobbin, Laboratory Director, online to answer questions after the session. This webinar is accredited through ACPE and is worth 1 CE credit hour. There is an evaluation and questionnaire link on the presentation handout. This handout is found on the lower portion of the GoToWebinar toolbox under Handouts. CE credit is applied following completion of the questionnaire and evaluation. For those unable to attend the webinar live today, we are recording this session and will include the recording and evaluation link in a follow-up email. Questions may be submitted throughout the webinar using the GoToWebinar toolbox on the right side of your screen under the Questions tab. We will answer the questions submitted following the presentation. If we do not get to your question today, a list of questions and answers will be emailed to all attendees following the webinar. You may also contact ARL through phone or email. This contact information will be included in the communication email to all attendees following the webinar. Thank you for joining today's session.
Okay. Thank you, Amy, and thank you everyone for taking time out of your day to attend the webinar. Again, I'm Brian Kelly. I declare no conflicts of interest. This presentation is accredited for, as Amy said, 1 contact hour through ACPE, and it's a knowledge-based educational program for pharmacists and technicians. The expiration date is 1 year from today. For complete ACPE information, see the program materials. Okay. So today we're talking about constructing stability studies, BUD studies to start. So we are going to define a stability study. In case you are curious as to why we are talking about stability studies for a BUD, that's because USP 797 says the only way to achieve a truly valid BUD is by conducting a stability study. Okay. We're also going to talk about some of the other mechanisms that USP allows for establishing a BUD. We're going to look at some of the other building blocks for building a successful stability study and some of the influencing factors or thought processes that need to occur to design a well-designed stability study. And then to wrap up, we are going to provide several examples of what a stability study might look like from a design perspective. So before we get into stability studies or BUD studies, start with what is stability. Per USP 1191, in short, you want your products to stay the same as they were at the time of compounding. Okay. Notice this definition from USP says that that you need to have specified limits in your stability study. I think everybody, for the most part, understands that the limits on their potency, typically 90 to 110, 90% to 110%. You should reference monographs and make sure that's correct, but I think everybody understands that's a specified limit. But we're going to talk a lot about, in this presentation, about the other tests that people should include in a stability study, and you should think about having those limits specified for those tests before initiating the stability study too. So just for example, we'll talk about that pH should be included in a stability study. So what's the acceptance criteria for pH as it potentially changes throughout the stability study? Is it plus or minus 1 pH unit, for example? We'll talk a lot about that appearance can provide valuable information for a stability study. So have an acceptance criteria for your appearance test. Is it okay if the product turns slightly yellow over time, for example? Okay. So what is a stability study? I think it's important that everyone understands a stability study is an experiment. Okay. In this case, it's an experiment to demonstrate stability. You can't always predict the results of an experiment beforehand, but you can do the best you can to educate yourself and make sure that the stability study has a good design and is setting you up for success. So remember back to the scientific method from school. You ask a question, you do research, you form a hypothesis, you test the hypothesis, you evaluate the test results, all those things. And sometimes those things have to be adjusted. You change your hypothesis, you retest. So keep in mind that a stability study is an experiment. Okay? It's a complex process because there is many factors that come into play in a stability study. It's a well-designed stability study is not simply testing the concentration of the drug. Okay. So why conduct a stability study? To exceed the maximum BUDs or use periods as defined in 795 and 797. Again, as I mentioned on the objectives, 797 says the only way to get a true BUD is by conducting stability studies. Okay, product-specific stability studies. So just why would someone want to exceed the 795 maximum BUDs or 797 storage periods for patient benefit, right? Patients don't always want to come into the pharmacy for a new script. Pharmacists don't want to have to, go in the clean room and get in a bunny suit and things all the time to make sterile products. So the pharmacy can save time and costs by gaining efficiencies and making batches of products and storing them all on the shelf, which if the pharmacy is gaining benefits, then the patient can also gain benefits by reduced costs. So extending BUDs is a mechanism to benefit both the patient and the pharmacy. So I mentioned that stability studies are a complex process. That's because there are so many factors involved in a well-designed stability study. So again, I think most people understand that a stability study or BUD study looks at the stability of the active ingredient or the drug. I think I want everyone to understand that the container closure system also plays a role in a stability study. Just because a specific drug product is stable in a vial does not mean that same drug product would be stable in a syringe or an IV bag. The container closure system is important in stability. We're going to talk a lot in here about the value of the other tests. The other tests are really dependent on the dosage form, and the formulation's important. You have to think about the interaction between the drug and the excipients. For those of you that don't know, excipients are the other ingredients in the formulation. So those excipients may have an effect on the drug itself. Certainly can impact the stability study design and the validity of the stability indicating method. As I mentioned, exceeding the maximum BUDs or use periods allowed in 795 and 797, as you'll see, the two chapters take slightly different approaches. 795 has what they call maximum BUDs. They do say that these maximum BUDs are if you do not have stability data. Okay, so on in the chapter it says that the non-aqueous maximum BUD is the earliest expiration date of any drug, or 6 months, whichever is earlier. As you'll see, these non-aqueous have a significantly longer maximum BUD than aqueous products. That's because non-aqueous products don't have the water available to support life of microorganisms, whereas the aqueous products, whether it's an oral, it's 14 days maximum at cold temperature, or aqueous, these other aqueous products are no longer than 30 days. So again, if you want to exceed these maximum BUDs, you have to have stability information. As I said, 797 has a little bit different approach. As opposed to having maximum BUDs, 797 has these storage periods. So the storage periods are, as it says here on the slide, if you don't have a sterility test, meaning on that specific batch or lot, and you don't have BUD data for that specific product, these are the maximum storage periods. The storage periods are based upon the compounding risk level, and it's low, medium, or high. If you have any questions about what level of compounding you are doing, what risk level compounding you're doing, reference 797, it defines those. So it's based upon the risk level and the storage temperature. So basically, the lower the risk and temperature, the longer the storage period. The higher the risk, the higher the temperature, the shorter the storage period. Notice that some of these are down to the hour. So if you're compounding and storing the product at ambient temperatures, the time of day at which you're compounding is important. If you make the product at 9 o'clock in the morning on Tuesday, you can't use that product till 9 o'clock at night on Wednesday, right? It's a 24-hour use period. Okay. So that defines the kind of the USP use periods and their thoughts on the stability indicating studies. Stability studies being the only way to achieve truly valid BUDs. However, 797 does allow for other mechanisms to determine BUDs, which includes using literature. So the chapter does talk about that using literature to assign BUDs yields a theoretical BUD as opposed to a stability study that yields a truly valid BUD. Okay, so the chapter goes on to talk a lot about that using literature introduces a likelihood of error or inaccuracy in your BUD assignment. So, the more differences between the literature that you're using to assign your BUD and your product, formulation differences, container closure differences, storage temperature differences, storage conditions differences, all those things can increase the likelihood of error in your BUD assignment. Okay. One of the other things that's hard to judge when you're looking at literature for BUD assignment is the validity of the test method. Was the person that published the method, did they really know how to develop and validate a stability indicating method? We'll talk about those quite a bit, stability indicating methods. Also, using literature to assign BUDs may not be acceptable by some regulatory bodies. Some of the state boards are not allowing that anymore, and certainly the FDA prefers to have stability data, stability studies conducted. Okay. 797 also allows a couple other mechanisms for assigning a BUD, and that is to look at the product labeling in consultation with the commercial product manufacturer. Okay. So you've gone through the decision process and have decided that you are going to do a stability study, to get a truly valid BUD according to 797. So where do you start, right? You start with the foundation of a stability study. The foundation of a stability study is always going to be a stability indicating method. Okay. A stability indicating method is a specific type of analytical method. Not all test methods are stability indicating. Okay, so just because it's an HPLC doesn't tell you whether it's a stability indicating method or not. Right? I use HPLC as an example because it's the most common laboratory instrument. It's going to be used for testing the small molecule drugs that compounders most frequently work with. But we'll talk a lot about we'll talk some about the other methods, test methods, instruments that may be used for stability studies. So just briefly, stress studies typically have to be performed on the test, before the test method is labeled as stability indicating. Or if you don't understand what stress studies are, what I mean by that, we will get into that. And then the Stability indicating method needs to be validated for the exact product being tested. Validating for the API itself is not a valid test method for the drug product unless a substantial amount of laboratory work occurs to prove that that test method validated for the API or drug itself is valid for the product being tested. So, in short, a stability indicating method must be able to separate degradation products. For those of you not familiar, this is a chromatogram. This is the way that an HPLC presents the information it collects. Okay. So in this chromatogram, it happens to be for editate disodium. That's the peak on the right there, starting around the 15-minute mark or so. Just in front of that editate disodium peak is a peak labeled as the degradant. Okay. So a fair amount of laboratory work had to go into separating that degradant from the editate disodium, the drug in this case. Why is that important to separate degradants? Is because degraded drug may not have the same physiological effect in the patient as non-degraded drug, right? So if the degraded drug is not going to have the effect on the patient, you don't want to count that in your stability study. So your method needs to make sure you are separating degraded drug. Okay, that's the basis of a stability indicating method. So if you think stability indicating method, think separates degradants. Okay. Just to be fair, that was a nice, pretty easy chromatogram that I showed for editate disodium. Chromatograms are always not that simple and easy. So just to present what the laboratory staff may actually be dealing with in the real world, this is a chromatogram of alprostadil in a mixture. So this is trimix. If you notice, a lot of these peaks are really blown off the scale. We've zoomed way in to be able to see that alprostadil peak. So when you think about chromatography, don't always have that kind of super simple chromatogram in mind. They are oftentimes a lot more complicated than that. Okay, this is a real chromatogram for a trimix. Okay. So for a method to be considered stability indicating, again, remember a stability indicating method is the foundation of a stability study. So for a method to be considered stability indicating, you need to do forced degradation studies or stress studies. Stress studies, those are performed on the finished product. Again, methods are validated on the specific product that's going to be put on stability. So the purpose of the forced degradation is to create degradants of the drug. The drug itself can degrade, but also excipients can degrade. So the laboratory will ask you for some of your finished product. They will conduct these forced degradation studies to create these degradants, and then they develop a method that separates the degradants, just like I showed in the EDTA chromatogram. Okay, so I think people tend to confuse forced degradation and stress studies as a mechanism to determine stability. They do not. The stress studies or forced degradation, they can give you some insight into why a drug might degrade, but the purpose of the forced degradation or stress studies is to create degradants that may occur during the stability study, and then you develop a method that is separating those degradants that you created with the stress studies. Okay, so I'm talking about degradation methods or stress studies. The way those are conducted is that the finished product is subjected to pH adjustments. The pH adjustments are both with acid and base. So the intent of those is to force the drug to degrade by using pH, right? You should also do heat and humidity adjustments so that if the product degrades when exposed to heat, that we will force that drug to degrade and create the degradants. Also light and oxidation. light exposure and oxidize the drug product also. So that's how degradation is performed, again, to create degradants so that a stability indicating method can be developed. Okay, so here's a couple more chromatograms. On the top is, again, a nice clean chromatogram where the API or the drug peak is out there 25 minutes, kind of all by itself. No additional peaks showed up, and this was with the heat and humidity stress. So for this particular drug product, it's not, what didn't degrade when exposed to the heat and humidity conditions. Whereas when we expose that same product to UV light, all of those areas circled on that lower chromatogram are circling peaks that showed up from the UV light that didn't show up from the heat and humidity. So those peaks are degradants that occurred from the UV light. So it's important that when the stability indicating method is developed, that all of those stress conditions I just talked about, are used to create degradants in the drug product. Okay, so I wanted to show here, basically what we mean when there's interference in a stability indicating method. Okay, so if you look at the box, again, the chromatograms, if you notice up in the top right, that shows the full scale of the chromatogram. We are on the larger box here, we're really zoomed in, looking for interference. And so the black peaks represent the reference standards. These happen to be the reference standards for the paraben methods. So the black lines are the reference standards. They're nice and clean. Only the peaks for the parabens. The blue trace, blue line, was a sample that actually did not include any of the parabens. So if you see there within the circle, there's a couple of places where there's a blue line underneath the peak of the black line. That's what we mean by interference. So as I said, this sample actually didn't include any parabens, but if you didn't know, you may think that there were at least a couple of them included. So the test method would need to be further developed to separate that interference. But just wanted to give a visual here of what we mean when there's analytical interference. Okay, so there's a lot of opportunities for potential interferences. We've talked some about the drug degradants that occur as a result of the stress studies. It's important to also understand that excipients, just like what we showed there, was the preservatives. Something in the finished drug product was interfering with the preservatives. Well, the preservatives can also interfere with the drug. Okay, so preservatives also can degrade. So the excipients, preservatives, for example, they also have degradation products. So the stability indicating method needs to be able to separate not only the drug degradants, but the excipients and their degradants. There's also process-related impurities and known related compounds that can be present. So I just wanted to briefly touch on what I mean by process-related impurities. And so just in short, process-related impurities are leftovers from the manufacturing process. This is the synthesis pathway for phenylephrine. And so on the top left there, that's the starting molecule used in the process of creating phenylephrine. But as you see along the way, there's many intermediate products that are created. And when the phenylephrine is purified, there's potential for some of these intermediates or starting material to still be present. The analytical method needs to make sure you're separating those things. Okay. So I did want to show that how part of the way that you may make sure that those process-related impurities are not interfering is that USP does sell related compounds, and those are typically those process-related impurities. Okay. So once you develop once a method is developed that separates all those potential interfering factors, the method needs to be validated. So just kind of quickly going through some of the method validation parameters. You want to make sure your results are exact, so that's the method needs to be accurate. You want to make sure the method is repeatable, so that's precision. You want to make sure it's accurate over a range, that's linearity. Your validation may also include robustness, where you purposely make little changes to the method to make sure that the method still generates accurate test results. Ruggedness may have multiple chemists and instruments run it to make sure the same test results. Freeze thaw may also be included, and that would be included if the drug product actually is frozen and thawed. So I did want to talk about that the different drugs may require different test methods. As I kind of mentioned earlier, there is no universal method for stability. So I think people commonly think of HPLC. However, that may not be the appropriate method for the drug, that drug product. So in the selection process of what the appropriate method is, I think it's important that people understand the theory behind small molecule drugs. That is that, as I said, they're small molecule drug. Their structure is relatively simple compared to large molecules. So the theory behind the small molecule drugs is they work if they're present. So that theory applies to the patient, but it also applies to the appropriate test methodology selected in the laboratory. So the HPLC is the most common, with UV detection, is the most common methodology that a laboratory would use for the small molecule drugs that compounders commonly work with. However, there are many other instruments and detector options available for small molecules. Compounders are starting to get more and more involved with larger molecules, proteins, and peptides. Those drugs, because of the complex structure of the protein and how the conformation or folding of the protein is very important to its biological function, that same theory of if present they work, that doesn't hold true for those. So in choosing an appropriate test method, you probably going to want to choose a test method that measures the activity of the protein, not just whether it's there or not. So with the folding be very important if you're trying to measure that with an HPLC. HPLC is not likely going to pick up on the actual structure of the protein and figure out if it's still intact and has biological function. So so appropriate method selection is important when developing the foundation of your stability study, which is a stability indicating method. And we spent a lot of time on stability indicating method. It's important. Without a stability indicating method, you do not have a stability study. Okay. So once your foundation of your stability study is built, meaning you have a stability indicating method, you can start thinking about the other the other parts of your stability study. Think about this, the walls and the ceiling, the roof. What am I going to do to make this a complete stability study? So the other building blocks of a stability study are the other tests that should be included in the stability study. These other tests need to demonstrate the chemical, physical, and microbiological stability of the drug product. As I mentioned a couple times, I think people generally get the stability study includes the concentration of the drug or the stability of the drug. However, for a complete stability study, you need to consider all three of these properties: the chemical, physical, and microbiological stability needs to be demonstrated. So the other tests to include in the stability study should be based on the dosage form. In other words, the formulation. Okay, so different dosage forms and different formulations are going to have different tests. Okay, so going through the typical tests here that may fall into those three properties that I described, again, chemical, physical, and microbiological. The chemical, the drug stability or potency, I think everybody gets that. People may not think so much about preserved products. that throughout the stability study, you want to demonstrate that the preservative itself is also stable, right? The chemical property. Another chemical property is the pH. So pH should have its own acceptance criteria, but as I mentioned earlier, maybe plus or minus 1 pH unit, for example. But pH can also provide valuable information if one of the other tests run into problems, right? So if you end up with instability of the drug or the preservative, was it the pH changing over time that created that instability? So pH, again, is a good chemical property that you'd want to evaluate during a stability study. Physical properties, so the appearance. So think about pharmaceutical elegance of the product over time. Does it maintain its form and function? Think about phase separation of a topical cream, right? You get kind of a liquidy part sitting at the top. The patient is not typically going to mix that up as well as you do, so that may be important on phase separation. I mentioned color change earlier. Is color change acceptable? Something you want to evaluate during a stability study. Also, visible particulate. Okay, that can be something that would be detected during the appearance test. Visible particulate, not only a problem in and of itself in the case of injectables, but it could also give you some insight as to what was happening with your analytical test results. Okay. And then in injectable ophthalmic solutions, you want to think about subvisible particulate matter. Okay, so physical properties that should be evaluated during a stability study. Also microbiological properties. For sterile injectables, you want to demonstrate that the container closure has the ability to maintain a sterile environment, right? So as the product's sitting on the shelf, it needs to maintain sterility. And so closely related would be endotoxin. So again, want to demonstrate container closure integrity throughout the stability study. So for non-sterile preparations, there are also microbiological considerations. You want to think about microbial limits. That's basically a test to demonstrate how much microbial growth is present in your sample, and is that increasing over time and increasing over time to the point where the patient's no longer be using that product. And then I mentioned preservative on the chemical side. Also think about the preservative on the microbiological side. There's a USP 51, this is an antimicrobial preservative effectiveness test. Basically, that test demonstrates that the preservative is effective against allowing microbial growth to proliferate in the sample. So again, so the chemical, physical, and microbiological properties of the product should be evaluated during the stability study. To help with this test assignment while you think about your stability study design, USP Chapter 1163, Table 2 is a very helpful table that can help provide some direction as to what the appropriate tests are based on the dosage form. So the dosage forms are listed down the left side of the table, and then across the top are the various tests. So just wanted to put this out there as a valuable table resource that's out there to help with appropriate study design. Okay, so you need to figure out what tests you're going to do, to evaluate the physical, chemical, microbiological properties. So now you need to think about what kind of time points you're going to want to do on your stability study. So the time points would vary based upon need and estimated stability. So if you're not going to need to make a batch to sit on the shelf for a year or for the patient's use for a year, there's no reason to do a stability study that long. Earlier when I was talking about remembering the scientific method with having a question, doing research, developing a hypothesis. So before going into a stability study and selecting your time points, you should see what information is out there to help guide you in selecting the right time points. Okay. So a minimum of 3 time points is recommended. That's really a bare minimum. A lot of times people think about that they'll make a drug product, just keep it at the pharmacy and send it in 3 or 6 months later, and then assign their BUD based upon that single test result. The problem with that is it's very hard to make a conclusion about where the drug stability ended up when you have no evidence of where it started, right? So say at that 90-day time point, you send it into the lab got 95% potency still, or stability. Well, no one would know for sure whether the drug product started out at 120% potency, and so over the course of that 90 days or 6 months is actually degraded into the acceptable range. So you want to have a time 0 and a T-in at the end of the stability study, but 2 points makes a trend, but it makes a very weak one. So the more time points you can add in there, the more power your data is going to have. The other thing to think about, if you were only to do, my example, if you were only to do a time 0 and a time 90, but when you got to the day 90, it failed stability. Well, now you just spent 3 months waiting for a stability study, and you have no usable data as far as extending your BUD. Instability data can be valuable, but it's not going to help you with the BUD assignment, which was the point of the study. So you want to have enough time points in there where you can pick up on trending, and you have a fallback point just in case at some point in there you run into instability problems. So another thing to think about on the time points is that not all tests need to be conducted at each time point. You may be able to spread out some of the tests, meaning conduct them at every other time point or something like that. For example, endotoxin typically can be okay to just conduct it at the beginning and the end of the study. Again, the USP 51, preservative effectiveness, may choose to conduct that only at the beginning of the stability study and then conduct preservative potency throughout. Okay, so you have flexibility in the design of the study. However, they need to be, think quality by design. Think about scientific justification for the decisions you're making on what tests you're conducting at what time points and why, and what the impact of that is. Okay. So another part of stability study design would be deciding on whether or not you're going to conduct an accelerated arm of the stability study. So I think there's some questions out there as to what exactly an accelerated stability study is. I think some people can think that there's a way to perform a stability study over the course of a day or a week or something, and somehow that gets you a year stability data. That's just not the case. So accelerated stability data is storing the stability samples at a higher temperature and higher humidity. Thought process behind that is that that's more likely to degrade the drug product over time at a faster rate than the actual storage temperature. For example, if it's a product that's typically stored in the refrigerator, the accelerated storage condition would be at room temperature. Okay. So the accelerated data does provide data for temperature excursions. Say the patient took that refrigerated product at home, and instead of putting it in the refrigerator like they were supposed to, they left it on the counter, and they call you and want to know if the product's still good. The accelerated data can help provide that data. However, accelerated data, I have temporary BUD here. That is because accelerated data is a way to get your dating in a shorter period. Typically, double would be okay. So, for example, if you have 3 months of accelerated data, you can assign a 6-month BUD to it. However, you can't just depend on that data alone. You have to conduct the stability study at the intended storage temperature. that's why it's a temporary BUD, because you can use that BUD until your stability study is completed at the actual storage temperature, keeping in mind that BUD may end up changing based upon the test results of the actual storage condition stability study. So there is some risk involved in using accelerated data to assign a BUD. Okay. So is it acceptable to update BUDs in real time? Sure. Can be justifiable as long as the data is looking good. It's important to look at trending of the stability data. Again, that's another value add for having more time points, so that you have enough data to make decisions on whether or not you're going to update your BUD as you receive those test results, for example, at day 30, day 60, day 90. Right. Okay. So hopefully now you understand the foundation of the stability study is a stability indicating method. The rest of the stability study is all of the other tests that you want to do. Those are based upon the dosage form, the formulation, etc. So now we're going to take a look at some actual constructed stability studies. Okay. So here we're looking at a sterile injectable aqueous solution that happens to be phenylephrine, no preservative present. Okay. So we are going to look at during the stability study, going to evaluate the appearance. Again, that's a physical property. So again, looking at physical, pharmaceutical elegance, phase separation, color change, visible particulate, etc. If you notice in this study, it's a little bit front-loaded here with 4 time points occurring through the first 90 days, and then that 180-day time point there at the end. So total of 5 time points. The pH is also going to be evaluated. Again, remember that's a chemical property. So going to collect all that, both the appearance and pH data at all time points, along with the particulate matter. Another physical test, so make sure particles aren't forming over time. And of course, going to do the phenylephrine stability at each of the 5 time points too. Right? But sterility going to space that out a little bit. Just do the kind of beginning, middle, and end time points there as far as time goes. So you want the data to support the sterile environments maintained. And then the endotoxin, going to space those out even further and just do endotoxin at the beginning and end of the study. Okay, so here's a sterile injectable, again, aqueous with a preservative. So all of the same tests at all the same time points as the non-preserved, but going to add a couple of tests in here. And so for this study, the ascorbic acid was preserved with benzyl alcohol, so multi-dose products. So I'm going to evaluate the benzyl alcohol potency at each of the 5 time points too, to demonstrate that benzyl alcohol is also stable, right? So benzyl alcohol can evaporate through the container closure system potentially, so you want to keep an eye on its concentration. And then you've got USP 51 there at the bottom to demonstrate the benzyl alcohol is still effective throughout the use period. So spaced out the USP 51 preservative effectiveness test, but backing that up with the benzyl alcohol potency throughout. And so here's a non-sterile oral preservative without a preservative. So this is a meprazole prepared just in water. And notice in this study design, there's no pH because it's an oral product. That's the decision that was made, despite what I was talking about earlier about a pH going to provide a lot of valuable information about stability if some instability was observed. Also, particulate matter has been dropped for a couple of reasons. One, it's an oral product, but also it's a suspension. Right? Suspensions, by definition, contains particulate matter. So, also sterility and endotoxin have been dropped because it's a non-sterile product. But it is a non-sterile product. It is in water, so there's potential for microbial growth. So included USP 61 at each of the 5 time points to make sure that the microbes present in this non-sterile preparation aren't proliferating to the point where the product is not safe for the patient anymore. Here's an example of a non-sterile, again, oral aqueous suspension with a preservative. In this case, measured appearance and drug potency throughout, but also measured the pH. That was, understood the value of monitoring pH, even though it wouldn't be required on an oral product. But adding in the potency of the methylparaben, the preservative in this case, and again spreading out the USP 51 to just beginning and end. Sterile injectable solution and oil with a preservative. So, basically all the tests that we've covered with the appearance, particulate matter, potency of the drug, potency of the preservative, sterility endo, and the USP 51. In this case, there was no pH measurement, and that's for a couple of reasons. The pH in oil is not a required test from USP. Also, a lot of pH readers have a difficult time reading the pH of oils, but also this injectable is intramuscular, so pH may not be as critical there for the evaluation. Sterile ophthalmic solution. So because this is an ophthalmic, no endotoxin, and particulate matter has switched from 788 to 789, different criteria for the particulate matter for an injectable versus an ophthalmic. So again, quality design by design, choosing the appropriate tests for the drug product. They want to show here, this is a sterile injectable trimix with a preservative, just to illustrate the fact that it's a good idea to measure the potency of all drugs throughout the course of a stability study. So in this case, with 3 APIs, a considerable amount more testing throughout that study. Here's a synthetic antibiotic, sterile, cefazolin in this case. Point being is that cefazolin is a synthetic antibiotic, so HPLC is the appropriate test methodology. Again, reference your USP monographs for appropriate method selection. But other than that, routine study design there. As opposed to a natural antibiotic, like vancomycin. Again, reference your USP monographs, because vancomycin is a natural antibiotic where the structure is much more complicated. And, as I talked about earlier, that maybe the HPLC might not pick up on some of those changes, but the monograph does have a requirement to look at the composition of vancomycin. So vancomycin is not a single entity. The HPLC monograph has requirements to look and see the makeup of the vancomycin, but the actual stability of the vancomycin is measured by USP 81. That is a antibiotic activity assay. So, appropriate test method selection based upon the drug, and again, kind of bringing home the point that there's no such thing as a universal method for determining stability of all drug products. To that point, this is the Avastin stability program. A lot of compounders are repackaging Avastin. The FDA came out with a document for what tests they require on a repackaged biologic. So if you notice here, in this case, Avastin, again, a protein, not using HPLC. Instead, we're using an ELISA test method. The ELISA test method is meant to measure the activity of Avastin, but there are several other tests here that the FDA requires based upon the drug, the formulation, published information out there about concerns of the repackaged Avastin, and things they want you to evaluate during the course of a stability study. for a well-designed study. So that's it. I hope everybody had an opportunity to learn something there about a well-designed stability study, understands the foundation of any good stability study is a stability indicating method, and that you shouldn't just worry about the stability of the drug, but you should also think about the other chemical, the physical and the microbiological properties of the compound preparation. Okay. So we'll take a look here for the last 10 minutes or so, and go through some of the questions that we received. We will write up all of the questions and answers, whether we get to them on this, during this last 10 minutes or not. And we'll what do we send that out, Amy?
Yes, the questions and answers will be sent out in about a week. And if you hadn't had a chance to submit questions throughout the presentation, go ahead and submit those now. We're still collecting, and we'll get started.
Amy, why don't you take that? Are the slides going to be available?
Yes, so the slides are available currently right now as a handout in the toolbox on the screen that you see on your computer. We'll also include that as a link as well as the recording tomorrow.
Okay, so one of the questions we got was, when you look at stability data, does it matter which chemical wholesaler you use? So I'd say stability data is formulation specific. So depending on, you say chemical wholesaler, was that a pure powdered API, or is that a commercial formulated product? If it's a commercial formulated product, those commercial formulated products often vary in formulation. Just because it's the same drug doesn't mean it's the same formulation if it's a commercial product. So if stability, just like a stability indicating method is formulation specific, so is the stability data itself. So I'd say in general, if it's on an API and it's the exact same pure API, it's the exact same formulation, the manufacturer shouldn't mean that much of a difference. But if you're using a commercial formulated product, like a sterile transfer, that can certainly make a difference. Dr. Cupic, you see one you want to take?
This is an interesting question. Is stability indicating methods required for analyzing the potency of preservatives over time in lieu of the
antimicrobial
effectiveness test? And as Brian pointed out, when conducting a stability study, if you remember his table, it did include both the anti-preservative effectiveness test, microbiological, but it also included a test for the preservative itself, a chemical test by often an HPLC assay. So that would be included. You would have to demonstrate that in the stability indicating method that you can test chemically by HPLC for the preservative as well as so I would say you cannot do it in lieu of atomic anti-preservative effectiveness. You would need both of them, as he demonstrated that.
Well, ideally, chapter 341, USP chapter 341 is the chapter that provides a lot of test methods for those preservatives. In that chapter, it does allow for plus or minus 20% of the preservative. So a little bit wider window there for preservative acceptance criteria than what's typically allowed for the API. In an ideal situation, you have both the potency and the AET data. Vince, do you have anything to add on that?
No, I mean, it's fairly standard. One of the questions that sometimes arises, especially for things that have multi-preservatives in them, is do you get interaction? And so you may have, let's say, your preservative degraded into something and the other preservative degraded into something else. Are you getting any combination effects or impacts? Brian talked a little bit about pH. Sometimes these preservatives' effectiveness are actually impacted by the overall pH in which they're sitting. So if that's changed, you would not see that in the chemical quant of the preservative, yet you would see that when you ran the AET test. So it's unfortunately one of those it depends type of answers. Certainly it's allowable to run the AET and then do chemical testing, but there are a few questions that could be asked of you if you didn't have that AET at the end. So sometimes it's just a little bit about your confidence in the formulation, and how much exposure or risk you want to take with that BUD assignment.
One of the questions we got was, what about container closure testing on sterile injectables? Does it say preservative-free?
Yeah.
So the background on container closure integrity testing is the FDA put out a document I think a couple of years ago where it talked about that container closure integrity testing may be a better mechanism to demonstrate that the container closure system can maintain the sterile environment throughout the storage period. The reason why they said the container closure test may be a better alternative as opposed to sterility and endotoxin testing was because they stated the weaknesses of the sterility test. The weaknesses of the sterility test is that you're not testing every unit. Like you can't test every unit container closure throughout the stability study, or else you wouldn't have any of those containers left for any of the other products. So we kind of went traditional there with the stability study designs of demonstrating sterility and endotoxin throughout the period, as opposed to the container closure integrity, which that FDA guidance doc does say that the container closure integrity can be used in lieu of sterility testing.
I'll take a question. It says, as long as everything about a formulation remains the same, will the stability study results apply regardless of whether humans compound the product or a robot? The product itself doesn't know if a human or a robot made it. The guidelines, according to ICH, FDA, and USP, states beyond use date or expiry date needs to be determined using that appropriate scientific method is based upon evidence. And so the position is that BUD should be based upon scientific evidence-based studies, and regardless if it's made using a robot or a human, it needs to be conducted.
Okay, so we got a question that says, can stability data be extrapolated from higher concentration to lower concentration with respect to regulations? So typically what you'd want to do in that case would be to bracket. So you'd want to perform stability studies on both the high and the low, and anything that fell in between the high and the low, if both the high and the low were stable, anything that fell in between those would also be stable. But no, you can't just do the high or the low and extrapolate, kind of one way or the other. I think, the safest bet would be to bracket and do the highest and the lowest. There's some other questions here.
We just have about a minute for one question. There's a lot of redundancy, and we're getting to the end of the
we want to be cognizant of everyone's time here, so we will provide answers for all of the questions that we received in the Q&A document.
Yes, so we'll have that Q&A handout to all attendees within the next week. A follow-up email with the recording and evaluation link for CE credit will be emailed tomorrow. And we'll also include information about our upcoming Quality Compounding Summit. This is held October 5th through 6th in Oklahoma City. The continuing education event includes 9 hours of CE credit with speakers from United States Pharmacopoeia, International Journal of Pharmaceutical Compounding, Pro Pharma Clean Rooms, ARL, California Pharmacy Lawyers, and Empower Pharmacy. Thank you for your time today.